These are the main functions of the lymphatic system. First, it returns the leak fluid and plasma proteins back to the blood. The increased pressure at the arteriolar end of the capillary will cause plasma and all its dissolved contents to escape into the interstitial space. In humans, 20 liters of plasma is released into the interstitial space each day due to capillary filtration, but only about 17 liters is reabsorbed directly at the venule end. What will now absorb the remaining 3 liters of fluid is the lymphatic capillaries. It also returns about 75 to 200 grams of proteins per day back to the blood. Next, it protects the body against invading microorganisms via two ways: by physically digesting and destroying microbes and harmful substances through the process of phagocytosis, and via the immune response wherein the T and the B lymphocytes proliferate in response to contact with foreign microorganisms. Both these cells will function to destroy the pathogen as well as exist as memory cells, always on the lookout for pathogens in the process of immune surveillance. Last, it transports large molecules like fats and hormones towards the blood for circulation. The lymphatic system consists of two semi-independent parts, namely a series of lymphatic vessels that contain the lymph plus the lymphoid organs. This is the series of lymphatic vessels that aim to return lymph back into the circulation. It begins with the lymphatic capillaries, which are found around blood capillary beds. Lymphatic capillaries are also called terminal lymphatics because they are blind or close-ended thin-walled tubes composed of a single layer of overlapping endothelial cells. The interior overlapping edges of the cells seem to act as valves, forming openings that are 5 to 6 times bigger than vein openings, through which fluid can enter. However, they immediately close to prevent outflow. They also contain anchoring filaments that connect them to surrounding tissues. Lymphatic capillaries unite to form pre-collector lymphatic vessels, which in turn combine to form larger collecting vessels that pass through lymph nodes for filtration. The lymphatic collecting vessels are very similar to veins in that they are low-pressure vessels. However, they have thinner walls with more valves. They are also more numerous with less tortuous pores, and they all pass through the lymph nodes. The largest collecting vessels will now unite to form the lymphatic trunks. The lymphatic trunks drain fairly large areas of the body. They are named mostly from the regions from which they collect lymph. The major trunks include the jugular trunk that drains the head, the subclavian trunk that drains the upper extremity, the bronchomediastinal trunk that drains the thorax, the intestinal trunk that drains the abdomen, and the lumbar trunk that drains the lower extremity. All the major trunks will unite to form the lymphatic ducts. There are two major lymphatic ducts in the human body, namely the right lymphatic duct that drains the right side of the head, neck, thorax, and upper extremity. The rest of the body will now be drained by the thoracic duct. It is the largest lymph vessel in the body that originates in the abdomen at the upper end of the cisterna chyli, which is an elongated sac located in the right lumbar region of the abdominal cavity. It collects lymph from the two lumbar trunks and the intestinal trunk. Both the right lymphatic duct and the thoracic duct will drain into the subclavian vein. The right lymphatic duct into the right subclavian vein, whereas the thoracic duct into the left subclavian vein, where the lymph will be mixed with blood. The lymphatic system also includes lymphoid organs. Lymphoid organs are classified into primary and secondary. Primary lymphoid organs are the site where lymphocytes are produced, mature, and develop the ability to recognize self from non-self, so they can mount a normal immune response following exposure to a foreign body in a condition known as immunocompetence. And these organs include the red bone marrow and the thymus. Once immunocompetence is developed, the lymphocytes will now leave the primary lymphoid organs and go towards the secondary lymphoid organs, which are now the site where the lymphocytes will be exposed to pathogens, or what we call as antigen challenge, and where they will now transform into their final subsets. The secondary lymphoid organs include the lymph nodes, the tonsils, and the spleen. Let's discuss the primary lymphoid organs in more detail. The red bone marrow is located in the medullary cavity of the long bones of children and the flat bones and epiphysis of adults. Though they serve as the main site of white blood cell production, they also serve as the site for the maturation and development of immunocompetence of a subset of lymphocytes known as the B lymphocytes. The B is for the bone marrow. The thymus is a flat, pinkish-gray, two-lobed organ lying high in the chest, anterior to the aorta and posterior to the sternum in the superior mediastinum. It is prominent in newborns and continues to increase in size during childhood when it becomes most active, after which it is gradually replaced by fats until only a scar tissue remains by adolescence. This is the model of the thymus in the laboratory. As you can see, it is a bilobed organ located anterior to the aorta in the superior mediastinum. This shows the cross-section of the thymus. The gland is divided into lobules. Each lobule contains an outer darkly colored cortex comprised of densely packed lymphocytes and a central lightly staining medulla that contains fewer lymphocytes. The thymus is the site for T cell maturation, which is regulated by hormones also produced by the thymus, which includes the thymopoietin and the thymosin. Next, let's discuss the secondary lymphoid organs. Lymph nodes are small ovoid bodies found at intervals in the course of the lymphatic vessels. The lymph node is enclosed in a fibrous connective tissue capsule, and like the thymus, it is comprised of an outer cortex and an inner medulla. The outer part of the cortex is dense since it contains aggregations of lymphocytes organized as nodules. Each nodule contains a circular structure known as a follicle with lightly staining germinal centers that contain mainly B lymphocytes. The deeper cortex contains the paracortical region that is filled with T lymphocytes. In the medulla, there are medullary cords that contain lymphocytes and macrophages, as well as spaces called medullary sinuses. This is now the model of the lymph node in the laboratory. As you can see, the lymph node is enclosed by a fibrous connective tissue capsule represented by the beige outer portion you see in the model. It is comprised of an outer cortex and inner medulla. The capsule sends extensions into the interior of the node, known as trabeculae, that divides the cortex into the capsule, known as the follicles. If the follicle has a central lightly colored area, that area is called the germinal center, which contains mainly B lymphocytes. The inner cortex is now termed as the paracortical region that contains mainly T lymphocytes. Within the medulla, at the center of the node, are dense lymphatic tissues called medullary cords that is not well illustrated in this model. It contains B cells that produce antibodies that flow out of the lymph node via the lymph fluid. These cords also contain numerous macrophages, which cleanse the lymph fluid before it exits the node. The lymph enters the node via numerous afferent vessels with narrow lumen, and they exit the lymph node via a single efferent lymphatic vessel that has a wide lumen. The main function of the lymph node is to filter lymph. Lymph nodes are arranged into five main groups. These are the deep cervical nodes that filter lymph coming from all the lymphatic vessels of the head and the neck. In the armpit, you will find the axillary lymph nodes that filter lymph from the upper extremity, the anterior chest wall, and the abdominal wall above the umbilicus, and the back above the iliac crest. In the thoracic part of the trachea and the bronchi will be another group of lymph node known as the tracheobronchial nodes, which contain some of the largest nodes that filter the lymph from the lungs and the heart. Now, anterior to the abdominal aorta will be the pre-aortic lymph nodes that filters lymph from outlying groups of nodes draining the gastrointestinal tract, the liver, pancreas, and the spleen. Finally, found in the groin area will be the inguinal lymph node that filters all the lymph coming from the lower extremity, the external genitalia, and the anterior abdominal wall below the umbilicus. Next is the largest lymphoid organ known as the spleen, which measures about the size of a fist. It is located on the left side of the upper abdomen, just underneath the diaphragm, near the tail of the pancreas. This organ is not essential to life. It is composed of a red pulp that is known as the graveyard for aged and defective blood cells, especially the red blood cells, and a white pulp composed of dense lymphatic tissues arranged around arteries. This darkly colored round area is the white pulp, and the surrounding red area is the red pulp in this cross-section of the spleen. The red pulp is where old red blood cells and platelets are destroyed. Hence, it also serves as reservoir for erythrocytes and thrombocytes. Whereas the white pulp is the area that has immune function by filtering blood. Our last lymphoid organ is the tonsil. Tonsils are incompletely encapsulated masses of lymphoid follicles supported on a connective tissue framework. They contain invaginations forming blind-ended structures called crypts. The crypts are where bacteria and particulate matters are trapped. They are usually located in the walls of the respiratory and gastrointestinal organs. 6 are found in the pharynx, and 2 groups are in the intestinal tract. Those found in the pharynx form a ring known as the Waldeyer's ring of lymphoid tissues around the throat. 3 are located in the nasopharynx, namely the nasopharyngeal tonsils in the roof of the nasopharynx. When they become inflamed, they become known as the adenoids. Next will be the pair of tubal tonsils found posterior to the opening of the eustachian tube into the nasopharynx. The other three in the oropharynx include the paired palatine tonsils attached to the sides of the walls of the oropharynx and the single lingual tonsil on the posterior one-third or the base of the tongue. Meanwhile, the ones located in the intestinal tract includes the Peyer's patches and the appendix. This is the laboratory model showing the different tonsils making up the Waldeyer's ring. This represents the nasopharyngeal tonsil located at the roof of the nasopharynx. The tubal tonsils will not be seen in this model since it is being covered by this fold of mucous membrane near the opening of the eustachian tube. The nasopharyngeal tonsil and the tubal tonsils primarily function to filter the air that passes through the area. Now, this represents the lingual tonsil located at the posterior one-third of the tongue and at the sides of the wall of the oropharynx will be paired palatine tonsils. The lingual tonsils and the palatine tonsils will filter the air, food, and water that passes through the oropharynx. This images shows the tonsils that can be visualized through the oral cavity if the patient widely opens the mouth. This represents the pharyngeal tonsil. This is actually very hard to see unless you have an instrument used by ENTs. Whereas this represents the palatine tonsil on both sides of the oropharynx. This is actually the tonsils that you usually complain of when you have difficulty in swallowing. Tonsils act as the first line of defense against ingested or inhaled pathogens by trapping them from the air that we breathe and from the food and water that we ingest. Again, the nasopharyngeal and tubal tonsils will mainly filter the air that passes through the nasopharynx, whereas the palatine and the lingual tonsils will mainly filter the food, water, and air that passes through the oropharynx. Peyer's patches are large, isolated clusters of lymph nodes found in the ileum. They trap and destroy pathogens in the area, preventing them from reaching the intestinal wall. This image in letter A shows the top view of the Peyer's patch, and B is the side view showing aggregates of lymphoid follicles on the antimesenteric side of the ileum, surrounded by abundant blood and lymphatic vessels. The last lymphoid organ is the appendix, which is a thin tubular sac that sits at the junction of the last part of the small intestines known as the ileum and the first part of the large intestines known as the cecum. And this is located in the right lower quadrant of the abdomen. Since this is a blind sac, its function is still unknown. Mediating humoral immunity, this image represents the antibody molecule, also known as the immune or gamma globulin. As you can see, it is Y-shaped, and it is comprised of two light chains and two heavy chains linked together by disulfide bonds. The amino acid sequence in the top of the Y varies greatly among different antibodies, hence it is known as the variable region. It is also the region that binds antigens. Below the variable region is the constant region that determines the mechanisms used to destroy antigens since it contains the complement binding site and the binding sites to different cells, especially the leukocytes. This table shows the five major classes of antibodies based on their constant region and immune function. They are arranged in this slide from most numerous to least numerous. So just remember the acronym GAMPE to remember which is which in terms of plasma percentage. As you can see, most of these antibodies are monomers, that is, they contain only a single globulin unit. Only IgA contains two globulin units as a dimer, whereas IgM is the largest antibody as a pentamer with five globulin units. You have to remember the main function of each of these immune globulins. IgG is the only antibody that can cross the placental barrier. It can fix complement and is the predominant antibody secreted in the secondary immune response. IgA is found mainly in all body secretions, including milk, and it mainly functions to protect body surfaces. IgM is the most efficient complement fixator because of its size. Aside from being found dissolved in plasma, they can also be attached to B cell surfaces where they act as antigen binding receptors. IgM is also the predominant antibody secreted in the primary immune response. IgD is the antigen binding receptor bound to B cell surfaces with an important role in the activation of B cells. And IgE are usually attached to mast cells and basophils and are involved in allergies. So this ends our laboratory discussion for the lymphatic and the immune system. Thank you for listening.
Do you have any questions on the topic? Any questions, people? Questions? Okay, if none, do you have